Welcome to this lecture on hybrid modeling. My name is Moritz from Stosch and I'm Chief Innovation Officer at DataHow. At DataHow, we provide services and solutions for model-based process development, in particular to the pharmaceutical industries, but also other industries. Why am I presenting you this hybrid modeling content? Well, hybrid modeling has been at the center of my career from very early PhD days on until now in my position at DataHow. I also co-edited a book on hybrid modeling in the process industries. And it has really been my conviction that hybrid modeling can offer many benefits and that if people know about it, they can use it in a much better way. And so this is also what I would like to convey to you in this short lecture and in the following scripts. Now let me first of all show you in which environment we're working. On the y-axis, you see the amount of data, and on the x-axis, you see the value that could be derived if data was available. Now, if we look at the Amazons and Googles, you'll see that personalized marketing has a high derived value. Of course, if they show you what you want to buy, you're more likely to buy it. So there's more value, right? And the amount of data is gigantic. Every time you browse, you generate data. Every time you search for something, you generate data, right? The next area is the area of optimized pricing, and this you might, for instance, experience if you look for flights. Flights are adapted, the pricing is adapted based on your searches, and it could ultimately go as far as offering butter at different prices to different people. There's also the area of a personalized workout, and here, for instance, you're generating data every time you're running and you're using some kind of watch or device in order to track your progress. There's a lot of data generated, maybe it's not as much as for marketing, but of course the value that can be derived from that data is not as significant, the economic value. Now, where are we in the process industries? We're down here on the bottom right. There could be a lot of value, but the amount of data that we have is not so high. And that is because data is expensive to generate. Every experiment costs a lot of money. Every analytics costs a lot of money. And so when we generate data, we generally need to be much more carefully how we generate it, such that it is informative. But if we had that data, we could derive great value. Now there's something in our industries that the other industries don't have, and this is knowledge. So what is the impact of incorporating knowledge? Let me give you one simple example just to make this more clear to you. So imagine we have a variable x which is impacting on a variable y, right? You fill, for instance, water into the tank and you're impacting on the height of the water in the tank. Now, if you want to understand how changes in x impact on y, you need to do experiments, right? Without any knowledge about what is going on, you will do a couple of experiments at different x's, and you will measure what happens in y. And then you will try to fit some kind of model to your observations. So you will try to predict, you will try to find a model that describes how x impacts on y. This is a very daunting exercise, and in fact you then also need to figure out what is the right model. Now, if you already have knowledge available, imagine, for instance, you know that the relationship is linear, then you need much less experiments. So how many experiments would you need in this case? 2, right? You would need 2 experiments. 1 to figure out the slope, 1 to figure out the intercept. And of course, the further you place these experiments apart, the better are your parameter estimates in principle, because the prediction interval, the confidence interval on the parameters becomes much smaller. Now, this means that if you incorporate knowledge, you have the chance to reduce the number of experiments and the data requirements, in fact. And it also means that if you incorporate knowledge, you can go beyond what was tested before experimentally, right? You can go to arbitrary x values and you know exactly what the y value is. And this is why knowledge matters and why combining what you know with machine learning tools will get you beyond the pure capabilities of machine learning and AI. So what does that mean for our industries? If you look at the other industries, the domain knowledge that is available is relatively low. Of course, you know that if you work out more, in principle, you improve more, and there's some heuristics around it, and the same is true for personalized marketing and optimized pricing. But physics, really underlying models, are only available in our industry. There's material balances, there's energy balances, momentum balances, there's knowledge about chemistry, biology, and so forth. So use that knowledge and try to incorporate it smartly into the models. And I want to just show you for a biochemical process example what the impact of integrating knowledge can be. So here we look at the production of a vaccine against whooping cough, and this is accomplished with an organism called Bordetella pertussis. Now this organism needs to be grown, and in order to grow it we need to provide it with substrate, with food, right? And the two substrates that are important for Bordetella pertussis are lactate and glutamate. And if you provide it with the substrates, then ultimately it will grow, meaning every cell will grow, it will double, right? So there's two things that we are interested in. Substrate consumption, this is the rate at which Bordetella pertussis consumes the substrates. And biomass growth, the growth of Bordetella pertussis. This is the rate at which the organism grows. Now let's look at experimental data. What we see here is the change in concentrations of lactate, glutamate, and biomass over time. In black, we have the measured experimental values. In red, we have a model without knowledge. In blue, we have a model with knowledge. And you can see, if you look at the different experiments, that in the beginning, substrate is relatively high, then it is consumed, right? The substrate concentration decreases. And for biomass, of course, we see the opposite. In the beginning, it is low, and then biomass really grows. Now, we've trained these models on training data, we've validated them on validation data, and you can see that there's literally no difference between the model predictions. But there will be a difference in a second. And before we look at the differences, let's look at the model. Now, coming back to the schematic that we had before, we can very easily describe the model, we can easily describe the process by looking at the model. So in fact, we have a batch process, and we can now describe the change in substrate concentration, lactate, by this very simple material balances. So we have dlac, which is the change in lactate, d signifying the change, over dt, the change in time, right? So when does lactate change over time? Well, if it is consumed, right? So there's a reaction which is accounting for the consumption of lactate, rlac. And we can do the same for glutamate, really. There's a change in glutamate if glutamate is consumed. There's a change in biomass if biomass grows. You get the drift, right? And now, just using a bit more rational, we can, for biomass, incorporate further knowledge. We can say that it is every cell that is growing, right? So growth is cell-specific. And this is this term mu that we have here. It's the specific growth. And we multiply with the concentration of biomass. And in fact, we can use the same rationale for substrate. So you see here the consumption of glutamate, V-glu, is multiplied by the amount of cells. So every cell consumes glutamate. And of course, glutamate can also only be consumed if glutamate is present. So this is why we multiply with the glutamate concentration. So that means that the change in glutamate depends on the specific substrate consumption rate, the cells, the amount of cells, the cell concentration, and the glutamate concentration. And the same can, of course, be done for lactate. So the first model is the model which we call the model without knowledge, and the second is the model with knowledge. And in fact, in these balances, we know everything but the biology, right? And what is the biology? Well, the biology is the rates here. If we wanted to express these rates explicitly, we'd have to go down and write how they depend on substrate concentrations or how they depend on temperature, pH, and so forth. But we're not really interested into that. We're just interested in modeling the process, and therefore we just want to approximate these reaction rates or specific reaction rates with a machine learning model. And this is what we do. So you can see here this machine learning model on the right-hand side. It's a neural network. And it has as outputs our rates, our growth rates and our substrate consumption rates. And as inputs, it sees essentially the process inputs. Now, we can do the same thing for the model with knowledge. The only difference is that instead of predicting the absolute reaction rates, we have the specific reaction rates here, right? The specific growth, the specific substrate consumption. But it sees exactly the same process inputs. And you might ask yourself, why all the buzz if the models were equally performing well on the training and validation data? Well, all the fuss because if we change the initial substrate concentration slightly, right? We reduce it here, as you can see, by 5 millimolar. Then all of a sudden, the model that has no knowledge incorporated can no longer predict accurately the evolution of the substrate concentrations. And even worse, right? It independently predicts that biomass will still grow. And you can see really here the impact of incorporating knowledge in describing accurately what is happening in the process. Now, how can we harvest this? And this is the second example on an industrial E. coli process that I want to present you. So similar to Bordetella pertussis, we now have E. coli, and we typically use E. coli to produce recombinant proteins. So you insert some snippet of DNA into the E. coli, and this snippet of DNA encodes for the protein that you want to produce. And you need, of course, to provide E. coli with the right conditions, meaning E. coli has to feel well. That means temperature needs to be fine, pH needs to be okay, and you need to provide it with something to eat, right? The U feed here. And if you find the optimal conditions, then E. coli will start to produce eventually. So in order to find the ideal conditions, what people typically do in industry is a design of experiment, right? DOE. So they vary each factor systematically by itself and also in combination and in nonlinear ways, such that you can figure out at which condition do I get the most product. Now, condition here refers to each of these spheres that you can see. Now, let us for a moment look at the process in more detail. We have, of course, a process evolving over time. And here you can see a typical biomass profile evolving over time. Now, this is a fed batch process, right? From a certain moment on, we start feeding with U-feed so that biomass grows further and that it starts producing. In fact, we also need to indicate to E. coli when it should start producing. And we do this with an inducer. So we add a compound, IPDG for instance, that signals E. coli that it should start producing the product. And this is what E. coli does. It then starts producing the product. And the specificity of this product here is that this product is accumulating inside of the organism, right? So you have a capacity limit. All right, so this is the evolution of the process for one experimental condition, for one process condition, right? Now, if you do this design of experiment study, you have multiple conditions. And by checking multiple conditions, you can figure out for the end, this is where you're interested in, how the amount of product changes in function of the changes in condition. So why would you use hybrid modeling? Well, there's huge advantages, as you will see in a second. So how do we model this process? As an engineer, you of course will write down the material balance. So let's jump to the material balance of biomass. We had that before for Bordetella, right? And it's very similar here. You just see that we added this D times X term here on the right-hand side. And in fact, this term is explicitly known. It's the dilution term. So we add feeding to the reactor. And because we add something to the reactor, we dilute the numbers of cells, right? It's a different whether you have 1,000 cells per 1 liter or 1,000 cells per 2 liters. So if you add a liter of whatever, the concentration will be lower, even so you have the same amount of cells there. And this is what this dx term is accounting for. Now, for product, as I said, we have a product which is accumulating inside of the cell. And so we look at the specific product, the product per cell. And we look also at the formation of product per cell. This is this VPX. And as I said, product will only be formed if inducer is present. So we multiply with the inducer, and this is really a variable between 0 and 1, 0 being no inducer, 1 being inducer, right? Now, in these equations, in these material balances, we again don't know the biology, right? This specific growth and the specific product formation rate. And here we again plug in a neural network that essentially understands or describes how our specific rates vary in function of the parameters that we're interested in, temperature, pH, and the new feeding. But it also considers how these rates change in function of how much product is already inside the cell and what the biomass concentration is. The cool thing is, we can very easily describe how changes in parameters, in temperature, pH, and so forth, impact on the profile, right? Impact on the process behavior. And you can see this here for two examples, for two run examples, where we have, again, with the points, shown the measurements. We have in the black line the prediction of biomass, in the gray line the prediction of a specific product. And you can see that the model describes the behavior of the process very accurately, despite having different conditions on the left and on the right hand side. Okay, that's fine, right? We have exactly the same result as the statistically designated experiment. But we have a result in addition. We can describe evolution. And in fact, what we can do is we can vary the initial time, the induction time, by using the model without losing validity of the model, because time is explicitly accounted for. And more important even, and very easy to grab, we can, as we can describe the evolution, check when we should harvest. And we can see here that at 32 hours, we already have the maximum product that we can get. So the total process is 40-something hours long. And that, of course, means you can save almost 15 hours of process capacity. That's a huge value. So that means we can not only describe the impact on the process parameters that we were interested in on the process behavior, but we also get additional factors that we didn't need to do any experiments for, right? We can describe the impact of changing inducer time. We can describe the impact of changing harvesting time. That's phenomenal. Now, when I presented this to the people at GSK that I was working with back then, and this was during my postdoc time in Portugal still, they said, okay, if you can describe how the process will evolve if we have temperature static, now can you also use the model to describe what happens if we do step changes in temperature, pH, and the feeding? And I said, well, it's a dynamic model. Of course, it can describe this. So they performed an experiment, and I performed the predictions for this experiment with the model that I just showed you. And what you can see here on the bottom part is the changes in temperature and pH over time, and also the changes in feeding rate over time. And on the top part, you can see the profiles of biomass on the left and specific product on the right. And in gray, you can see the respective predicted specific rates. And you can see that the model fits the experimental data very well, despite the fact that it has never seen such changes in the process conditions. And what is more interesting to see, you can also see how the specific rates change in function of the process parameters. So you get this additional insight of what's happening with the cells, and this will really allow you to first design the process more rationally and also make scale up in a much more rational fashion. Now let me wrap it all up. What I hope you got from this lecture is that you can leverage your data with knowledge, and you have a model that typically provides you with more understanding, that allows you to reduce the data requirements, and both on the quantity and quality side. And reduction in experiments really means reduction in cost and reduction in time, and reduction in time in the pharmaceutical industry is one of the most important factors. And these models also allow you to extrapolate, right, to search in regions that had not been covered in the data before. So it really has attractive properties. At this stage, and before leaving you to the script and whatever you're seeing next, I want to bring on a disclaimer. And that is, hybrid models do not deliver value by chance, but by design. And in fact, this is true for all models. So before you start your modeling exercise, always ask yourself, what do I want to do with the model? And what properties, what benefits do hybrid models for this specific case provide me that other models cannot provide? And once you answer these two questions, your rationale is clear for setting up a hybrid model. There's no one to stop you. With this, I'd like to thank you for your attention and wish you all success in your learning journey. Cheers.